Good afternoon, everybody. Many thanks to the organisers for the invitation to speak at this meeting. It's a pleasure to be back in Chicago for another PRRS symposium and the CROAD meeting. I was asked if I could actually speak about perspectives on the development of the global effort for disease control. And I'm focusing on the research, the global research effort in relation to controlling providing the tools for controlling animal disease. So I'm going to basically talk about the background to the global effort on research coordination that I've been involved in over the last 10 years, the International Research Consortium on Animal Health that has evolved, and the partners and governance structure of that organisation. I want to look at addressing priority challenges. I may comment briefly on how we arrived at those challenges. I'm going to then look at system solution maps. And then there's something which is taking up a lot of attention at the moment to do with antimicrobial resistance and alternatives to antibiotics. And then perhaps touching on preparing for future challenges, which follows on nicely from things Paul said a few minutes ago. New challenges are constantly rising. You know, whatever it is, 13 years ago, Hendra appeared by surprise in Australia. A related virus, pigs, humans, appeared in Southeast Asia, Nipah, 1998. Blue tongue virus appeared in North Africa and into the Mediterranean about 1999. You got West Nile virus across here about 1999. SARS appeared in 2002, and despite there only been a few cases, it actually caused billions of dollars of economic loss. Of course, completely surprised, blue tongue virus 8 appeared in Northern Europe in 2006. African swine fever the following year appeared in Georgia, in the Caucasus. Pestipity ruminants appeared in China in 2010. PED appeared in China about 2010. Possibly was around a year or two before that, because the comments at meetings back about 2009 suggest it may have been circulating before that. Schmallenberg virus, of course, appeared in Europe again, totally by surprise, in 2011. And more recently, MERS has appeared in the Middle East. So you can see the challenges are many, and they're constantly, new ones are constantly appearing. Of course, there's all the existing disease challenges as well, the endemic diseases that we live with, respiratory disease of pigs or whatever. Re-emerging diseases, especially in relation to drug resistance, things that we had under control, especially in ruminants, anthelmintic drug resistance, has caused problems with controlling those diseases. Threats from epizootic diseases, ever present, foot and mouth disease, African swine fever, classical swine fever, whatever, avian influenza. And I couldn't show the avian influenza outbreaks on the last map because they've just been so frequent. Extension of the range of vector-borne diseases, new strains of organisms constantly emerging, new disease challenges. So we can't all do everything, hence the need to work together. Back about 2005, I got involved in setting up a collaborative working group concerned with the coordination of animal disease research across Europe. But of course, as you can see, the problems tend to be global. So the European Commission decided it would be good to actually have a global network of the major research funders or program owners on animal health. And that's where the STAR-IDAZ project arose from. And that's the acronym for Global Strategic Alliances for the Coordination of Research on Major Infectious Diseases of Animals and Zoonoses. And that was a global initiative to coordinate or to address the coordination of research programs at international level in the area of animal health, in particular infectious diseases. We had, in that network, 25 partners from 18 countries who had signed the grant agreement, and they were partners to the project. But then under it, we also set up regional networks, bringing in smaller players, both So we had a regional network for the Americas, one for Asia, Australasia, and towards the end we also set up one for Africa and the Middle East to complement the existing network that I mentioned earlier on in Europe, the Collaborative Working Group on Animal Health and Welfare Research. The scope was emerging major infectious disease of production animals, including aquatic animals and bees, zoonosis, diseases of wildlife, where the wildlife basically served as a reservoir of infection, either for humans or for production animals. Achievements of that network, because it was a 4-year project, it now moves forward as a self-sustaining network. But the achievements of the project, the 4-year project, was we had a database of research publications, useful starting point to actually identify who is carrying out work on the various diseases. We also developed a research programs database, looking at what was going on in the different countries in terms of what capacity there was, what was the major focus of the research activities. We also looked at research needs at global, regional and industry sector level. And this is where I mentioned identifying priorities. We asked our partners to tell us, number one, what were for every sector, pigs, poultry, dairy, beef, small ruminants, what were the 6 most diseases that they were spending the most money on? In other words, where were they spending the resources? The second question was, what were the 6 most important diseases affecting or causing economic impact on the various livestock sectors? And the third question was actually, what were did they see as the emerging threats? And on that, we identified our priority diseases and issues related to things like antimicrobial resistance or drug resistance. We also looked at instruments to enable cooperation, clustering and partnership. And we developed a long-term strategic research agenda, and I'll come back about that on that towards the end of my talk. But probably most important, we actually ended up with an extensive network of about 50 countries, network of research program owners and program managers in about 50 countries that we could interact with. The European Commission was then actually interested in moving into high level. I mentioned that STAR-IDAZ now moves forward as a self-sustaining network under an agreed memorandum of understanding. But the European Commission was interested in moving it to higher level. They looked at what was happening on the human health side. There was a number of big initiatives which were referred to as International Research Consortia, and the suggestion was that we move forward in that form. We basically developed similar structure to what was happening in relation to human rare diseases. So to be a partner in the IRC, an organisation had to have a minimum level of investment in research on priorities over a 5-year period of about $10 million. But groups could come together as consortia. They had agreed to delivery targets, agreed to coordinate or align the funding to deliver these targets, so members using their own funding procedures and whatever. And agreed to share results, bearing in mind the need to protect intellectual property. But we're looking at options whereby we will interact with the pharmaceutical industry, and hopefully they will see us as honest data brokers and disclose to us what they're doing with confidence that we won't actually tell their competitors. So far, of the original STAR-IDAZ partners, 22 partners from 14 countries, including one international research organisation, the International Livestock Research Institute in Nairobi, Kenya, one charity, the Bill and Melinda Gates Foundation, the European Commission, and 3 industry partners have signed the letter of intent to participate. It would be actually quite good to see the 3 industry partners that are involved are actually from the pharmaceutical industry. So it would be good to actually get livestock industry partners involved as well. And those 22 partners over a 5-year period have got a combined research budget of about $2.5 billion for the priority areas that we're talking about. So you could argue that there's quite a bit of scope there for coordination of activities. I've listed here the signatories. You will see there is one group that joined as a consortia from Belgium because there's a group of smaller organisations. But anyhow, the presentations will be available, so you can check it later. Right. The overall objective is to coordinate research at international level to contribute to new and improved animal health strategies for at least 30 priority diseases, infections or issues. Now, this was agreed at a meeting organised by the European Commission in Brussels in October 2015. The deliverables were to include candidate vaccines, diagnostics, therapeutics, other animal health products and procedures, key scientific information tools to support risk analysis and disease control. We're especially interested in, sorry, can I go back? Yeah. So we're especially interested in hard deliverables, vaccines, diagnostics or therapeutics. The structure is basically shown here. The top box is the IRC Executive Committee, which is basically the funding organisations or programme owners. We have established a scientific advisory committee of 16 people from different backgrounds, virologists, there's pig virologists there, virologists, immunologists, bacteriologists, parasitologists. So 16 different people, 16 people on it from different countries around the world. Don Knowles from USDA-ARS is one of the members of the scientific committee. We plan setting up working groups on a range of diseases or in the process of setting up ones. I've shown here in red ones particularly relevant to those working with pigs. African swine fever, influenza, coronaviruses, PRRS, porcine respiratory disease complex, the bacterial component of it, and foot and mouth disease. And I've shown in purple others that are also related to your interest. Immunology, vaccinology, diagnostics, which we heard mentioned earlier on, innovative anti-infective approaches. I'll come back to why that may be particularly important to you. Animal genetics and genomics in relation to disease control. We will constantly keep adding others to it as it moves on. The coronavirus one is an interesting one because I could see us being interested in developing coronavirus vaccine platforms for things like poultry or pigs. Infectious bronchitis and virus in poultry is constantly changing. New strains come along, so one could, if one had a vaccine platform, one could change the spike protein genes depending on what strain was moving around. And perhaps similar type things could be done for porcine epidemic diarrhea with the coronavirus vaccine platform for pigs. The European Commission, what I could have mentioned, of course, is that actually sitting off to the side, we've now got a secretariat that underpins all of that, and I lead the secretariat. The partners involved in that are definitely the Department for the Environment, Food and Rural Affairs in London, which is where I work. BBSOC, which is the Research Council in the UK, one of the research councils in the UK. OIE, and we've got somebody based in OIE, and that allows us to basically interact with their disease experts around the world. And also CABI. Now, some of you may know the Commonwealth Agriculture Bureau as it was, but they do the CAB abstracts, Index Veterinarius and Veterinary Bulletin they used to publish. And the International Federation for Animal Health, which is now called Animal Health Europe, are the partners in the Secretariat. And the acronym for it is CIRCA, Secretariat for International Research Consortium on Animal Health. And our activities will be to basically work with the scientific committee in establishing working groups on those priority diseases or issues. work with some of the experts in the field in developing gap analysis. Where does research need to be focused? And then developing roadmaps, and then mapping current funding activities against those roadmaps and see where new research money needs to be spent. We will also look at how research programs can be aligned to address the gaps. We'll work and try and help to mobilize resources, bringing together the funding bodies and helping to identify funding opportunities. And I mentioned earlier on the involvement of the pharmaceutical industry, so we'll work closely with people in the big pharma, looking at knowledge transfer to bring innovation to market. The working groups that we have in the process of setting up, we'd like to get one moving forward on PRRS now. Bovine tuberculosis is being set up or has been set up. Brucellosis we're ready to take forward. African swine fever, helminths including antibiotic resistance, and vaccinology. And others that we want to move forward with, foot and mouth disease, vector-borne diseases, and coronaviruses. And then we'll come back to looking at the others as well. Of course, in a couple of those areas, there are already extensive networks of researchers. So, for example, in foot and mouth disease, there's the Global Foot and Mouth Disease Research Alliance. On African swine fever, there's GARA, Global African Swine Fever Research Alliance. So there's the GFRA on foot and mouth disease. There's Global African Swine Fever Research Alliance. On those, so we will work with those groups in developing the research gap analysis, and then what we will do is actually map current funding activities against those research gaps. I mentioned vaccinology. Of course, important is actually deciding what type of vaccine one needs. What are the specifications of the vaccine? What are we trying to do? And perhaps a good way of actually looking at how activities should be focused would be by developing system solution maps. And this is an early draft of what we're trying to do. So basically, you look at the various actors in the system. So in relation to vaccine development, is everything from the pathogen and identifying its virulence factors, out to things like the regulators, society. So all of that will help to identify what would be acceptable as a vaccine. And in developing roadmaps, we're looking at this sort of model for actually developing, so looking at various leads. So, for example, on the very right-hand side, you could argue that the need in relation to PRRS is a vector vaccine that is, covers the different strains. So basically, the need is a new vector vaccine covering the different strains, for example. And then under that, identifying the challenges. What are the solution routes? And then mapping the projects that are addressing those, or they're involved in addressing those challenges. But of course, that all depends on, so there are dependencies, that all depends on, if you're talking about a vector vaccine, identifying what is the most suitable vector. It also depends on identifying what are the most suitable antigens to get expressed by that vector. So you can see that takes you back to the next lead. Maybe the next step behind that is not just one box, it could be 4, it could be more. So we'll work with some people in the research community in developing a roadmap for the delivery of whatever type of vaccine is needed. And then the whole thing will be summarized here. So the research question, our needs, the needs. So it would be, for, as I said, perhaps for PRRS, it might be a vector vaccine. What are the challenges, the solution routes, the dependencies, projects? And then at each level, we work out that type of system. So it's putting structure to the research needs and working all the way back from what we need to control disease back to basic research. A few years ago, I interacted with some of the people in the PRRS community looking at what the research needs were. they're shown in this spreadsheet here. I've since updated it with information from Jones' paper, a colleague's paper, on, which is really excellent review of the immunology relating to PRRS. And I'm going to re-engage with those people who we spoke to a few years ago to validate what we've got here. And I'd previously looked at developing a roadmap in relation to PRRS, and I thought, right, PRRS vector vaccine makes a lot of sense. I'm not personally convinced about an attenuated vaccine, but I could be persuaded otherwise. Perhaps a kill vaccine might also be really useful, depending on adjuvants and whatever. But it seemed to me that looking at the research gaps in the spreadsheet, there were a lot of research needs which were fairly fundamental science. In other words, what was happening in the macrophages infected by the virus. So looking at gene expression, both viral and macrophage gene expression under different in vivo environments. I'll just follow the bottom line. On the basis of that, you could identify the stealth mechanisms or identify virulence mechanisms. A lot of that has already been done. And on the basis of that, genetically modified organisms could be generated, and perhaps that could give rise to a new PRRS vaccine. But we will revisit this and try and put it into the structure that I've shown earlier with the different boxes. Right, vaccines. Back in, I think it was April 2015, the OIE brought together some people in Paris to look at where vaccines were needed that would significantly reduce the need for the use of antibiotics. So where were vaccines needed to have greatest impact in reducing the use of antibiotics? And the people they brought together for that meeting in Paris decided to just concentrate on those industry sectors where populations are treated rather than individual animals. In other words, pigs, poultry and fish. They ignored the ruminants. I believe they are now going to actually come back and look at the ruminant situation. But of course, in ruminants, you tend to treat just individual sick animals with antibiotics. In pigs, poultry and fish, the perception is that they're treated as populations. So the areas where they identified where vaccines were most needed, and they're not all bacterial, because some of them are viral. But anyhow, Streptococcus suis, Haemophilus parasites were mentioned. Other specifically respiratory pathogens, Pasteurella multocida, Mycoplasma pneumonia, Actinobacillus pleuropneumonia, porcine reproductive respiratory syndrome virus, swine influenza virus. Moving on, E. coli. And in Wiener's finishers, again, E. coli, Lawsonia intracellularis, Brachyspira, and rotaviruses. These were the ones where they actually suggested that vaccines were needed. I think Bob mentioned earlier on about funding opportunities. I think along with some of this, there would appear to be opportunities for research funding. Because it's getting a lot of attention at the moment, and significant resources are being put into it. Right, in terms of longer-term challenges, sorry, I should finish up. Under STAR-IDAZ, I mentioned earlier on, we developed a long-term strategic research agenda. In the Americas, we actually looked initially using scenarios developed by the Canadians. In Africa and the Middle East, again, we did driver prioritization and looked at the impact of those. There was a separate study in the Mediterranean. In Europe, we actually did a lot of work on driver prioritization. What is driving change? What are the likely disease impacts of that? And then we developed scenarios and then looked at backcasting to a preferred future. This is where we want to be in 30 years' time. How do we get there from where we currently are, taking into account the challenges identified in the scenarios workshops? And on the basis of that, we developed a long-term strategic research agenda. I'll just mention some of the drivers that were identified: population size, density, and demographic change, including movement of people. Of course, related to that is also people becoming more affluent, and therefore need for animal-based proteins. Markets increasing. Economics of farming, including profitability and competitiveness. There's been too much pressure, actually, downward pressure on food prices, which has been driving intensification and whatever. Balance between economy, ecology, environmental impact, animal welfare. There's all these issues come in. Climate change, perhaps less important in relation to pigs, but I could be wrong. Pathogen evolution, including drug resistance. Specific topics that were mentioned: diseases or challenges. Improved understanding of the role of wildlife in the spread of disease. That's probably less important in relation to intensive pig production, but obviously important in relation to intensive poultry production and influenza. Vector-borne diseases, they seem to be somewhat on the move. In relation to the pig sector, perhaps African swine fever falls within that category, but then with ticks, but then maybe wild boars are more important. Antibiotic effectiveness and availability. And as that word availability becomes important, moving forward, will antibiotics be as available as they currently are? I think the World Health Organization had something to say about that fairly recently. So that is one of the challenges for the industry moving forward, is actually the availability of treatment. Disease introductions, including transboundary animal diseases, I mentioned that earlier on. Improved food safety, gut health. That kind of links somewhat to antibiotic effectiveness and alternatives. Antibiotic resistance, new diseases. Anyhow, we're going to revisit this. So under the Global Network, we will set up and conduct further Foresight activities and update all of this. Of course, there's the other side. Technology can actually be a driver of change, but technology can also help with addressing the challenges. Things that were identified as being important were diagnostic tests, express methods, pen-side tests, and things like that. Vaccine development, new genetically engineered vaccines. We need a lot of new and more rapid response vaccines. Alternatives to antibiotics, antimicrobials. Alternative methods to control vectors, biosecurity, systems-based approach research. So that's the contact details.